is Brooklyn Prejean, and I'll be reading slides 21 through 30. Characteristics of antigens must be perceived as foreign. What are antigens and what do they do? Antigens are engulfed, eaten by APCs. Antigens elicit immune response. Epitope, 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 part of the antigen is binding to the T/B cells. Example, whole microbes or their parts. What antigen characteristics make it a good immunogen? Elicit a lasting immune response. Proteins are more immunogenic than repetitious polymers, carbs, bacteria, LPS, glycoproteins, etc. Large MW moles as or better. Different types of antigens, alloantigens, and superantigens. Why don't I have the rest on here? Alright. Section 13.4, Step 3, Antigenic Challenge of T cells and B cells method. Learning outcomes. Summarize the process of T cell activation and list major types of T cells produced in this process. Diagram the steps of a B cell activation and list the types of B cells produced in this process. Step 3, Antigenic Challenge of T cells and B cells. As T cells react with APC and recognize the antigen/MHC complex, the one that fits the best is selected, and it becomes active and differentiates into effector T cell and memory T cells. Then the colonial expansion ensures enough cells to activate immune response. Effector T cells. These types, T helpers, help everyone, T regulator cells, put a cap on things, and T cytotoxic assassins, and 3 types of memory cells. Each subtype has its own memory cell. T helper is further subdivided into Th1 and Th2 cells. No functions also. T helpers help other cells by producing cytokines. No need to know the names of individual cytokines. T cytotoxic cells, assassins, kill body cells affected with virus or cancer. T regulatory cells put a stop when the immune response goes too far. As B cell recognizes the antigen, it can recognize it without presentation, but it also can be presented via T helper. It is also activated and differentiates into B effector cells, including plasma cells, makes antibodies, and B regulatory cells suppresses T cells activity, as well as B memory cells. When clonal expansion or proliferation happens of many clones of the same cell, which was the best at binding the antigen. Section 13.5, Step 4.1, the T cell response. Learning outcomes. Describe the main functions of major T cell types and their subsets, including T helper, T cytotoxic, and T regulatory cells. Explain the role of cytotoxic T cells in apoptosis and list the potential targets of this process. Diagram an antibody binding antigen and list the possible end results of this process. List the 5 types of antibodies and important characteristics of each. Draw and label a graph illustrating the development of a secondary immune response.